Mendel's law of independent assortment states that genes for different traits are inherited independently of each other. A dihybrid cross is a type of genetic cross that examines the inheritance of two different traits at the same time. Each trait is controlled by a different gene, and each gene has two alleles, a dominant and a recessive. When two plants that are heterozygous for both traits are crossed, it's called a dihybrid cross, like crossing heterozygous G's and W's by heterozygous G's and W's. To solve a dihybrid cross, we use the FOIL method to find all possible combinations of alleles each parent can pass on. To find the allele combination, we use the FOIL method. First, outer, inner, last. First combination is dominant G and dominant W, the first set of alleles in each genotype. Next is dominant G and recessive W, the outer set of alleles in each genotype. Then, recessive G and dominant W, the inner set of alleles next to each other. And the recessive G and recessive W, the last alleles in each genotype. Now we build the 4x4 Punnett square using those combinations from each parent. These are the 4 gametes from each parent. Keep adding each gamete to the Punnett square. We'll use this to find all possible offspring combinations. Here's the outcome of both parents, and we populate the Punnett square like so. First, you have the dominant G and the dominant W being placed along the outside of the 4x4 Punnett square. Both parents are heterozygous for both traits, therefore each will have the same outcome when we FOIL. If only one set of gametes are shown, do the following. Here's the outcome of both parents, and we populate the dihybrid cross like so. Take the dominant G and the dominant W from the first position and place it on the side of the Punnett square. Going back to the original where I gave you both parent gametes combinations, you take the dominant G and the recessive W and place them on top, and it will also be the same on the side of the Punnett square. Third, you'll have the recessive G and dominant W along with the recessive G and dominant W along the side. Lastly, you'll have both recessives on the top and side of the Punnett square. Next step is to fill in the Punnett square to find the genotypes of all possible offspring. We now fill in each box of the genotype from combining one allele from each parent. Slide and drop each allele. Dominancy matters as the dominant letter will always go first within the new genotype. Keep the different alleles separated, all G's to the left and all W's to the right. In the second top position, you slide and drop, paying attention to what letter is dominant. Third position, fourth position, and I'll model the fifth position. Continue adding alleles to the genotypes. Each genotype gives us a phenotype. For example, heterozygous G's and W's means green and wrinkled. We use dominant traits to decide the outcomes. The next step is to identify the phenotypes based on the dominant and recessive allele combinations. Now that the Punnett square is complete, we can label each box by phenotype based on the alleles. For example, any genotype with at least one dominant G and one dominant W will result in green and wrinkled seeds. Out of the 16 boxes, 9 show green and wrinkled phenotype. These have at least one dominant G and one dominant W. 3 boxes are green and round. These have at least one dominant G and 2 recessive W alleles. 3 boxes show yellow and wrinkled. These have 2 recessive G alleles and at least one dominant W. 1 box shows yellow and round. This is the only box with all recessive alleles, G's and W's. You could color code like so. Green is dominant G, red is wrinkled dominant W, yellow is recessive G, and round is recessive W. Here I marked all the dominant G, whether it be homozygous dominant or heterozygous. To get the green phenotype, the box only needs to have one dominant G. Next, I'm going to search for the genotype that is recessive G for being yellow. There are 4 boxes that show this. I put red to indicate the dominant W to have the phenotype of being wrinkled. We only need one dominant W, therefore it could be homozygous dominant or heterozygous. There are 12 boxes that show this. Lastly is the recessive W, or in this case homozygous recessive to have the phenotype of being round. There are 4 boxes that show this. Here I put all the phenotypes in place of the genotypes. Next, we're going to determine phenotypic ratios. How many out of 16 are green and wrinkled? What boxes show green and red? Count them up. There are 9 boxes that are green and wrinkled. How many out of 16 are green and round? Homozygous dominant and heterozygous Gs, and homozygous recessive Ws. If you count them up, there's 3. How many out of 16 are yellow and wrinkled? There are 3 yellow and red combinations. Out of the 16, how many are yellow and round? Lastly, there's only 1 homozygous recessive Gs and homozygous recessive W. When 2 plants that are heterozygous for both traits are crossed, it often results in a 9:3 to 3:1 phenotypic ratio. To summarize, the dihybrid cross looks at 2 traits at the same time. Using the FOIL method, we find possible gametes, fill in the 4x4 Punnett square, and determine phenotypes. When both parents are heterozygous, we usually see a 9:3 to 3:1 ratio. This supports Mendel's law of independent assortment.